Hello, my name is Haley Rhodes, and today we are going to do a presentation on aldolase and glycolysis and gluconeogenesis. So our objectives today are going to be looking at what and how aldolase functions, in addition to how it functions within glycolysis and gluconeogenesis, and looking at how aldolase is expressed. And then we'll have a quick summary at the end. So we can find aldolase in most living species on Earth. Aldolase is split into two main classes. Aldolase class 1 is found in animals and higher plant tissues. Class 1 aldolase are best classified by their lack of bivalent metal cofactors. Class 2 aldolases can be found in prokaryotic and eukaryotic and lower eukaryotic species. Class 2 aldolases require bivalent metal cofactors. There are three types of class 1 aldolases. Type A is primarily found in muscles and some in the brain. Type B is found in liver and kidneys. Type C is found in the brain as well. And we'll talk a little bit more about each of these types in a little bit. Aldolase is required to act as a catalyst in key reactions in glycolysis and gluconeogenesis. The substrate for aldolase is D-fructose. The three types that are found within mammals are composed of 4 subunits, each subunit containing 360 amino acids. The structure of each subunit contains a beta barrel that is surrounded by alpha helices and connecting loops. Active sites are in constant communication, and if activity within one subunit is affected, the entire protein is not capable of function. So we can look at the aldolase picture up on the screen. You see the 4 subunits. It's kind of hard to tell, but they're pretty similar in structure. And again, there are 4, which is important. So in order to appreciate how aldolase is used in glycolysis, let's take a moment to walk through the process. So starting in step 1, hexokinase, which all of the enzymes used through glycolysis are listed here in blue. And then the substrates are going to be listed above and below each enzyme. So in step 1, hexokinase will phosphorylate glucose by taking a phosphate group from an ATP molecule and binding it to glucose, making a molecule known as glucose 6-phosphate. In the second step, we can see the enzyme phosphoglucose isomerase acting upon the glucose 6-phosphate molecule. This enzyme will isomerize its substrate into fructose 6-phosphate. While glucose 6-phosphate and fructose 6-phosphate have the same molecular formula, they have different atomic arrangements, making them two different molecules. So on to step 3. PFK represents the enzyme phosphofructokinase. PFK will use an ATP molecule to add another phosphate group to the F6P and form fructose 1,6 biphosphate. So bi is 2, so it has 2 phosphate groups added onto it now. So next on to step 4, which is kind of what we're going to focus on in both glycolysis, and then we'll come back and talk about it in gluconeogenesis. But we'll see that aldolase is going to split the fructose 1,6 biphosphate molecule into dihydroxyacetone phosphate, or DHAP, and glyceraldehyde 3-phosphate, or 3-GP. Step 5 shows triose phosphate isomerase. Changing the DHAP into G3P in preparation for step 6. Step 6, we will see glyceraldehyde 3-phosphate dehydrogenase, a hydrogen molecule, or take a hydrogen molecule from the G3P and then add it on to a NAD, or a nicotinamide adenine dinucleotide, to form NADH plus H, which is your hydrogen ion. Secondly, in step 6, adds a phosphate group to G3P, forming a 1,3 biphosphoglycerate, or BPG. In step 7, we see phosphoglycerokinase transfer a phosphate group from BPG onto an ADP molecule. And that ADP molecule turns into ATP. So the BPG molecule then forms 3-phosphoglycerate, or 3-PGA. In step 9, we see phosphoglycerate mutase move a phosphate from the 3rd carbon to the 2nd to form 2-PGA. In step 9, Aldolase removes water from 2-PGA to form PEP, or phosphophenol pyruvate. In step 10, pyruvate kinase is going to transfer a phosphate group from PEP to an ADP molecule to form forming pyruvate. And then again, that transfer of that phosphate group to ADP turns it into ATP. So in glycolysis, we're able to, we only net 2 ATP molecules because we used 2 ATP molecules in order to get through the whole process. We used 1 ATP molecule in step 1 and 1 ATP molecule in step 3. So even though your step 7 and step 10 each showed +2 ATP, again, you have to subtract the 2 that we used in steps 1 and 3. So adding and subtracting it all together, you see that you net 2 total ATP molecules. So let's take a closer look at the role of aldolase in step 4 of glycolysis. Again, aldolase is responsible for cleaving the fructose 1,6 biphosphate molecule The two molecules that are formed at the end of the reaction are isomers, known as triose phosphates. The molecules are glyceraldehyde 3 phosphate, or G3P, and dihydroxyacetone, or DHAP. When taking a look at the reaction, aldolase will cleave or break the bond between the third carbon and fourth carbon. So the dysfunction of aldolase can occur in different individuals. Some individuals have a mutation that can cause a deficiency of aldolase type B. This deficiency can lead to the inhibition of glycolysis. If aldolase is not present, then an accumulation of fructose 1-phosphate will occur, which can be toxic. In addition, this can lead to the depletion of ATP and inorganic phosphate within the cell. On the opposite side, the upregulation of aldolase is often seen in tumors. So let's now look at how aldolase is used in gluconeogenesis. So gluconeogenesis is the process of making glucose from non-hexose precursors. Non-hexose precursors are going to include molecules such as glycerol, lactate, pyruvate, and glucogenic amino acids. In some ways, glucogenesis is glycolysis in reverse. One of the biggest differences is that is the enzymes that are used within gluconeogenesis, and there are 4 different enzymes that are used in gluconeogenesis, and then in glycolysis. So those 4 are going to be pyruvate carboxylase, PEP carboxykinase, fructose 1,6-biphosphatase, and glucose-6-phosphatase. And the reason for these 4 is that these 4 are located within reactions that have no reverse to them in glycolysis. Another important thing to note is that glucogenesis can only occur in certain types of cells because these particular enzymes are only located within those cells. So when starting with pyruvate, gluconeogenesis will begin within the mitochondria. So first, pyruvate carboxylase will carboxylate the pyruvate to form oxaloacetate. So we can see this kind of at the bottom of the screen. So pyruvate to oxaloacetate. This process is going to require the use of ATP. So if you remember, at the end of glycolysis, we had made 2 ATP in that area. So we are using 2 ATP molecules to pretty much drive the process backwards. So this process will require the next step occurs in the systole, which is the cytoplasm. Here, PEP carbokinase is used to decarboxylate the oxaloacetate to form PEP. And then steps 9 through 4 are reversed using the same enzymes that are within glycolysis. So if we go back to our glycolysis slide, we can look at steps 9 through 4. So we had made PEP in our last step that we talked about in gluconeogenesis. So after PEP, Aldolase is going to then turn PEP into the 2-phosphoglycerate. Phosphoglycerate uses the enzyme phosphoglycerate mutase to go back into 3-phosphoglycerate or 3-PG. And then 3-PG is going to use phosphoglycerate kinase to go into 1,3-biphosphoglycerate. Then that will go into the glyceraldehyde 3-phosphate dehydrogenase enzyme will turn the biphosphoglycerate into 3-GP, and then your triose phosphate isomerase enzymes will turn 3-GP and DHA-DP into those two, and then aldolase comes in again and in step 4 and turns it back into fructose 1,6-biphosphate. So if you notice here, the arrow only points down, meaning that with gluconeogenesis, we're going to use a different enzyme here. So I'm going to go back to our gluconeogenesis slide. So now that we are here, so in order for fructose 1,6-phosphate to revert back to fructose 6-phosphate, the enzyme fructose 1,6-biphosphatase is needed. Fructose 6-phosphate is then changed into glucose 6-phosphate by the enzyme phosphohexose isomerase. Lastly, glucose 6-phosphate is dephosphorylated by glucose 6-phosphatase to form glucose. And again, that was a one-way reaction in glycolysis. So luckily, as you're going through the processes of gluconeogenesis and glycolysis, most or many of the enzymes that you have in each are typically very close to your previous molecules that are being made. So kind of makes it a little bit easier to remember them all. So when looking at aldolase and how there are different isoforms that are expressed, to better understand how it functions in different areas. So aldolase type A is typically highly expressed in heart tissues, as well as the tissues in the esophagus and the brain. You'll also see within this graph, and whenever we go to the next 2 slides, that almost every tissue that is listed here has type A aldolase within it. So that's pretty important that we can almost find type A anywhere in the body. But again, it's mostly expressed in the heart, esophagus, and brain. There are mutations that can occur within this particular enzyme, and they're often associated with glycogen storage disease. This disease is associated with hemolytic anemia. In addition, disruption of this type can lead to various types of cancer as well. And again, the various types comes because aldolase A is found in so many different types of cells. Next up is aldolase B. Is definitely not expressed in nearly as many type of tissues as type A, but type B can primarily be found within the kidneys, liver, and small intestines and duodenum. You can see that there's small amounts also found within the gallbladder, and then a very teeny tiny amount found within the colon as well. So mutations of type B are associated with hereditary fructose intolerance. This mutation changes the shape of the tetramer, so the tetra is 4, so it changes the whole shape of the enzyme, which is not good. So a lack of function of type B aldolase can result in a buildup of fructose 1-phosphate within the liver. The buildup of this molecule in the liver can be toxic and can cause the death of liver cells. So type C, aldolase is primarily expressed within the brain, and in the brain it's particularly expressed within the hippocampus and the Purkinje cells. So in addition, there's also a little bit of a higher number than everything else of heart and fat tissue expression as well. And again, there's small amounts of many of them, but comparing these to type A is found predominantly in almost everything. So lastly, let's take a look at type B and different transcription factors that play a role in the gene expression of type B. So there are 4 different transcription factors that play a role in type B expression: HNF1, HNF3, C, EPB, and then DPB. Aldolase type B is primarily found within the liver. So most of the studies that have been completed on type B transcription factors are done typically on liver cells. So one study looked at hep G2 cells, and it was noted that HNF1 and DPB were strong trans activators, meaning that they liked and wanted to increase gene expression, while CEPB was also a trans activator. However, it had a weaker expression. And then HNF3 function as an antagonist to HNF1, DPB, and DPB. So HNF3 decreased gene expression. So whenever we're looking at aldolase, aldolase plays a huge role within glycolysis and gluconeogenesis. Without aldolase, or excuse me, without aldolase or having mutations within aldolase, you can almost stop glycolysis in its steps and have large amounts of the fructose 1,6 biphosphate, which is not good in many cells and can be very toxic in many different cells. And again, it's expressed in many different types of cells. Type A had lots of different types of cells, and the heart was a big one there. Type B was primarily found within the liver and kidneys, and then type C was primarily found within the brain. Again, that was in the hippocampus and the Purkinje cells. And then lastly, we talked about type B transcription factors, which had HNF1 and DPB, which were high trans activators, and then your HNF3 was a antagonist to those two. So thank you for taking the time to watch this presentation. These are all of the references I used throughout the presentation. Some are for pictures, some are for different resources that I used. Thank you and have a great day.